Thank you so much for listening and being here today. My name is Mahnoor Aziz Khan, and I'm a student of bioengineering in Sabancı University, Istanbul. Right now, I am in Maastricht University in the Netherlands doing health sciences, and I want to share one of my projects, which I did last year. It's called High Precision Pathogenicity Prediction of PKLR Mutations. So it's basically a Python-based predictive tool which helps detect for now related to PKLR mutations, whether they are pathogenic or benign. Okay, so I would like to start with the introduction to my disease. So it is basically PKD. It is a rare genetic disorder. It is called pyruvate kinase deficiency, which is basically due to the mutations of the pyruvate kinase enzyme, which is used in the glycolysis stage, or you can say the energy producing stage inside the cells of red blood cells and the liver. But basically, PKD is caused due to hemolysis in the red blood cells because red blood cells only use glycolysis as their energy source. And if there's any mutations in this enzyme, then basically you cannot convert pyruvate into ATP, which reduces the energy and ATP production, as well as reduces the red blood cells lifespan from 120 days, which is the usual or the normal, to just a few days. A little bit about the pyruvate kinase enzyme itself. So it is a tetrameric protein, so it consists of three domains, which is A, B, C, and also domain N, which is not entirely used in the study that I have done, as well as it has 574 amino acids. And according to the mutations I have studied, literature suggests more than 300 mutations, which can be present in the pyruvate kinase enzyme, resulting to PKD. And the gene is on chromosome IQ22. Okay, so what is the main objective of my study? It was to develop a computational tool which can easily identify if there are any mutations in specific missense mutations, which were the focus of my project. If the missense mutations on the amino acid chain will result in the enzyme being pathogenic or benign, because some of the mutations, even though they happen on the enzyme, they don't directly result in a pathogenic disease. If the mutation results in a pathogenicity or a benignness, they have very different outcomes. So if the mutation is pathogenic, then of course it will result in the disease. So the main thing was to make a computational predictive tool. So the first step of my Python prediction tool was to, of course, look into the literature. And according to the literature screening, I found out that there were different drivers such as thermodynamic instability, structural disruptions, and different functions of the allosteric and the active site, along with impaired kinetic efficiency, which can influence the enzyme dysfunction. Moving on to the method of how I made the Python predictive tool. As first, I did a literature scanning and found out the important things which actually do affect the enzyme. Just to be sure, I only focused on the missense mutations. Of course, there are other mutations like frame shift mutations and nonsense mutations that can affect the enzyme as well. But I personally focused on missense mutations in particular. So the data I gathered was related to the evolutionary conservation scores, the Gibbs free energy, the distance from the active side, and the distance from the allosteric side. So these became the four main features which I used in my Python training program in order to curate a ground truth table from which I can further train my data. And the analysis was related to polyphen and SIFT. So I compared it at the end, my results with the already available results in polyphen and SIFT. Okay, so the literature suggested that the extraction of the conservation scores, how I did it was I used the BLAST algorithm, and then I used it with the alignment of the multiple sequence alignment. On the right, you can see one of the radar graphs that I made for the beta domain. I also made them for the alpha domain and the C domain. And these were important because they were telling me, okay, in the 574 amino acid sequence length, which particular or the alpha domain is evolutionary conserved. It is because if amino acid is evolutionary conserved, then if there is a chance that there is a mutation, then it would result in pathogenicity. So all of these, and the distance from the allosteric side, the distance matrices, the Allosterian distances, all of them were important as features in order to predict whether a mutation at any position of the amino acid chain for the enzyme will result in pathogenicity or benignness. What I did was I got the results and then I compared them with polyphen and SIFT. So SIFT already gives a specificity of 0.4 and polyphen gives a specificity of 0.6. The dataset I curated and trained my Python tool on was of 126 variants, which I got from different websites as well as from PubMed, from ClinVar. This enhanced as a training set on which I trained my Python tool on. And at the end, the Python tool I developed was a truth-based Python tool. And at the end, I used this Python tool to have a Matthews correlation coefficient, F1 scores, and then the specificity and the sensitivity of the Python tool were also calculated by me in order to compare them with SIFT and polyphen. I made an ROC analysis, ROC curve. So you can see that the orange one indicates my PKLR tool, which has an AOC of 1, which shows very high effectiveness and very high productivity. The dotted blue one indicates the SIFT and the green one indicates the polyphen. So polyphen and SIFT have an ROCs of 0.7 and 0.8 respectively. So there's a little bit misalignment, but my tool gave a perfect alignment of 1. This is some of the snapshots from the Python tool itself. So basically, the PKLR tool asks you which position you want to check within your set, if it is pathogenic or benign. So you can put in, okay, if position number 23, will it be pathogenic or benign in your case? Then it runs through all the available 20 amino acids and then gives you a predictive score. And it tells you, okay, the number of predictions it has, how different they are. And it gives you not just for my tool, but already available tools from polyphen as well as from SIFT. But as you can see, the accuracy, sensitivity, and the specificity of my tool is really high. And it not only uses one aspect, such as ISIT distances, so the distance from the allosteric side or from the active site, but also it keeps in mind the evolutionary conservation scores, which were currently not present in SIFT and polyphen. In conclusion, there is a need for checking whether a particular mutation will be pathogenic or benign if it is changed or swapped with any other mutation, any other amino acid. So it was a very comprehensive way of checking whether a particular mutation will be pathogenic or benign not. With this, because this was a project that I did in my university, we were also able to convert the same idea and use it for many other genetic diseases, and they can be related to other genetic diseases which we have already available sequence of amino acids of, so we can check if we change one of the amino acid to any of the other 20, how will it affect the pathogenicity and the benignness. One important thing I want to add as well, which is currently the future aim, we also looked at the kinetic inefficiency and the Gibbs free energy, how it can affect how it is present at each amino acid level and how it can affect the pathogenicity and benignness. But in the scope of my project, it was a little too much because I had a limited time. But right now in the future, we are looking over how we can incorporate the kinetic efficiency of each amino acid along with the evolutionary conservation scores to check whether there will be an overall effect on the pathogenicity or benignness as well. Yes, if you have any questions, and thank you so much for listening to me. You can also ask my questions now, or you can mail me as per your convenience.